Neemt u plaats. Esteemed PhD candidate, the College of Deans of this university, represented by us, has read your PhD thesis and the propositions that go with it, and we are ready to hear you to defend it. Please take your seat. Does anyone in the audience wish to exchange ideas with the PhD candidate? If not, then I call upon Professor Jiskoot, Professor of Drug Delivery of the University of Leiden. Very welcome to you.
Thank you. Meneer de kandidaat, esteemed candidate, congratulations with the appearance of your very nice PhD thesis. Very important topic, drawing vaccines and proteins for stabilization. When I was reading your title and Drying Made Easy and then read your thesis, I sometimes got the idea that you took it a bit too easy. Also, when I was reading the second line of your thesis, I was wondering how you spray dry a promising technology. But I won't argue about the wording. I would rather like to go to the contents. And I would like to focus on the particle size, and not the particle size of the powders, which you characterized quite well, but the particle size of the API, which is either the monoclonal antibody or one of the viruses you worked on. And maybe I can start. How important is particle size in vaccination or in vaccine delivery in general?
Ho, clear opponent. Thank you for your question. I understand particle size is a very important component, especially for vaccines. That can give an indication about aggregation if it's increased, or the subvisible particle size, which is responsible for the immune responses, and there is a lot of literature available. In terms of why is it important, is that they can give an immune response, which is unwanted sometimes, especially in cases of antibody, infliximab, for example. Although that was not investigated, but I did read some literature on that. For our formulations with vaccines, we did see some changes, but they were not too big in terms of that we could say that it was changed during the drying to a substantial level.
That may be true, but when I go, for instance, to chapter 4, which is about the polio vaccine chapter, the only readout that I see is the de-antigenicity of the virus, and I don't see any data on the particle size. So don't you consider this as being important for virus vaccines?
We don't mention it in the manuscript here. We did check the particle size, which is around 30 nanometers, if I remember correctly. Initially, during our drying process, we didn't see changes, at least in the particle size. However, the major challenge that we were facing was the de-antigenicity was going to 0%. So if you have no de-antigenicity, it's a bad formulation. Slowly, when we started getting recoveries which were a bit higher, or considerably higher than what we were expecting before, we focused on that time being. We did analyze the particle size and didn't see, it's not reported in the manuscript here.
Okay, thank you. Let me move then to chapter 6, which is about a monoclonal antibody, infliximab, where you don't want particles, actually. well, you conclude that drying is a good way to stabilize this protein. Freeze drying a bit better than spray drying, actually. But when I go to page 136, 137, I saw to my surprise that after reconstitution, you see even visible particles. And you kind of ignore those. You filter the thing and you centrifuge some solutions to get rid of these unwanted particles. So I think this is a problem when you would have this formulation in a vial and you reconstitute it and you want to inject it and you see visible particles.
Indeed, it's a good observation that you made. We also did observe it. The reason to filter them was when the filtrate was taken out, it was analyzed if there was any protein content, and we didn't see a change after filtration in the protein content. And we assumed that this visible particles were dust, which could be during the drying process came in.
Well, there's many examples in the literature where there are subvisible and visible particles present, which is not noticeable by the protein content because the amount is so very small. And even 0.01% of the total protein content, which you won't measure as a difference, can give rise to millions of particles sometimes. So I'm not so sure whether this is not proteins. Did you try to analyze the particles or the things you spun down?
No, we didn't analyze them for any further.
Okay. So let me go, if there's some time left,
There is
to
time.
the particle sizes that you did analyze. Let's see. And maybe I will start with figure 1A.
Mm-hmm.
It's an SDS page, gel under non-reducing conditions, which is supposed to be a monomeric IgG with a molecular weight of 150,000. And even in the untreated material, I see 1, 2, 3, 4, maybe 5 different bands. So what's happening here?
Indeed, we also observed that, and we assume that these extra bands are a result of impurities, which were present already in the formulation. So
why don't you see them with the size exclusion chromatography? Because there you see one nice monomeric peak for the untreated material.
Yes, indeed. So we were not exactly sure what the nature of this material was. However, we did investigate, but could be the case that you could isolate these extra bands and check them through mass spectrometry. That wasn't done in this case, and we assumed them to be impurity, which came with the infliximab sample.
And how about the sample number 8, so lane number 8, I should say, which is spray dried in the presence of sucrose inulin? I see a very clear increase in one of the bands at about 100 kD and also a lower molecular weight band at about 40 kD or something like that. You don't comment on that. I think it's an important observation because it looks like something is going on there.
As I mentioned before, that we assumed it as impurities and we didn't comment on that. Indeed, if we really want to comment, we would need some data to back up that, and that could have been done with the MS, and that was not investigated in this study. So just saying it there was something is better than saying it with an analysis.
Okay, I can buy that. Maybe final question?
1 minute,
1 minute.
including the answer.
Including the answer. Okay, I will be very brief. You looked at small aggregates with SDS page and SEC, and you noticed but ignored the visible particles. There's a very large size range in between, which you did not look at. So how would you analyze the potential aggregates that are not seen by SEC and SDS page and by the naked eye?
Indeed, so the figure that you see here, that is figure 2A, which is actually the analysis of samples that were stored at a high temperature. I mentioned it on page 139, the analysis of the samples just after drying. We didn't see any aggregates.
Yeah, but you did not use methods which look at the larger aggregates. That's my point. So SEC measures aggregates until, well, maybe 100 nanometers, but from 100 nanometers and 100 micrometers, which is the subvisible gap, so to say, there may be a lot of aggregates. And typically we see those a lot in protein formulations.
Yeah, I understand your point. However, this analysis was not done, so I cannot comment further on that what was the nature of those aggregates if they were there.
Thank you very much for the discussion.
Thank you for your opposition. Then I call upon Dr. Hendricks, Pharmaceutical Technology and Biopharmacy Department in the University of Groningen.
Esteemed candidates, I would like to congratulate you and also your supervisors and your co-supervisors with the completion of this thesis. I think it's very nice work. You know, I worked with influenza vaccines and infliximab in the past, but not on polio and pertussis vaccine, so it was quite nice for me to learn about these vaccines. But I have a question about the aim of your thesis, what you state in page 12. And it was a bit intriguing because there you say, I use influenza vaccine as a model, and you don't mention that with the polio vaccine and the pertussis vaccine. So apparently these are not models, but the influenza is. Can you explain me why suddenly influenza vaccine is a model? You also mentioned that in chapter 3, by the way, so maybe you can answer.
Very clear opponent. Thank you for your question and the nice comments about the book. It's an interesting observation that you made with the model candidate vaccine. Model, in terms what we mentioned in the aim, is for the design of experiment study. So to understand how a vaccine is behaving during spray drying, we first were investigating the process parameters. And to check the process parameters, we need a candidate for that to optimize. And for that, we considered influenza vaccine. We in the past had, me myself had experience with drying it, so that is why I mentioned it as a model candidate vaccine. While the other chapters, we are not directly using design of experiment at every step of the chapter, and that's why I don't mention it as a model vaccine.
Yeah, but at the end, you also look at stability of the vaccine, and then actually it seems to be extremely stable. Because in all cases, when you store them for a long time at elevated temperature, you don't see any loss of antigenicity. So in that sense, it may not be such a good model in the way that other vaccine might be much less stable. And then the process conditions could lead to other results.
Absolutely correct. I also mentioned in the chapter that indeed the model is very nice to optimize the physical parameters of the particle size, the RMC, powder yield. However, due to low change in the antigenicity of the vaccine was a downside that you cannot model it, that how it will behave differently in different drying conditions. So it was too stable to, so that was a downside. We thought we would see some changes. I did mention in the discussion that the temperature range that I use for drying was very moderate, and it could be the case if you expand it to a bit higher temperatures, you might see some changes.
Yeah, but why do you want to go
that
to high
again is
temperatures?
a point that why in a real condition would you use such a high temperature just for a case to have a vaccine that will be unstable during drying? You would not like to waste your antigen in a way. It's not commercially thoughtful to use such extreme temperatures for drying. But indeed, that was a downside of the model.
Okay. Then I would like to ask a second question about chapter 3 actually, and it's about the particle size distribution. You're aiming for a particle size of 1 to 5 micrometer because that is the size range you want to have for pulmonary delivery. But then, to my surprise, you express the particle size as the X50 value, which is actually the median. So it means that at first sight, when I see, okay, I have a particle size of 5 microns, I think, okay, that's nice. But when you consider the X50 value, assuming Gaussian distribution, then you know that 50% of the particles will be larger than the 5 micron. So why, for example, you could also say, okay, I want, for example, 80% of the particles in the size range of 1 to 5. Why didn't you express it in this way? Why did you use the X50 value?
Okay. So apart from the X50 statement, which is in the table, I also mentioned the span, which includes the X10, X50, and X90, which is a clear indication of how your sample would look like in an overview. And Although that wasn't not really mentioned in the quality target product profile, we decided to just mention the X50 values. And indeed, what you want to see in terms of an in-a-label particle, you want that to be the aerodynamic particle size. We didn't measure it, which needs some further experiments, but can be always calculated based on the density of the particles.
Exactly, that was also my concern a little bit, because you mentioned that when you change the concentration of the trehalose in the solution to be sprayed right, it will affect the porosity, and thereby maybe not the geometric particle size distribution, but it will do with the aerodynamic. So how would it influence your results if you consider, instead of the geometrical particle size, the aerodynamic particle size distribution? Would it affect your results, do you think? And how?
I mean, if you change the concentration of the trehalose?
No, for example, if you have changed the concentration of the trehalose in the solution, and you get the same geometric particle size, then the aerodynamic particle size will change. So on first sight, I would say think they're the same, but they're not in terms of aerodynamic. So how would it affect the results of the conclusions of your study?
Indeed, if you change the concentration, if it not changes here, it will change the density of the particle.
Yep.
That indeed will influence the aerodynamic particle size. Either you measure it or you calculate it theoretically. So there will be a change.
Yeah.
And I think that should be also taken into consideration when actual model is used with the vaccine.
Yeah. Well, what you could have done is measure density with pignometry, and then you could. But anyway, thank you very much for your answers, and I give the word back to the.
Thank you for your opposition. Then I call upon Professor Hukride, Professor of Vaccinology, University Medical Center of Groningen.
Esteemed candidate, it's a real pleasure to see you defend your thesis today. We know each other already from your master time as a master student in the group of Erik Freiling, so it's really great to see you defending your thesis today, and I would like to congratulate you on finishing this very interesting thesis for sure. So you will not be surprised that I will concentrate with my questions on chapter 5, where I finally also see some in vivo results, which is more interesting for a vaccinologist, of course. However, to start with, I was wondering, you investigate in chapter 5 pertussis vaccine, and you choose the OMV pertussis vaccine for that purpose. Why that one? And would there have been other alternatives still sticking to pertussis vaccines?
Clear opponent. Thank you for the question and the comments on the book and, of course, the whole trajectory here. I understand your concern with the different vaccines in terms of the disease caused by pertussis bacteria. There are 3 vaccines available. 2 are traditionally used, whole cell and the acellular pertussis vaccine. But due to the strain adaptation and the waning immunity, the new investigated vaccine that we have at our institute is the outer-membrane vesicle vaccine. There is another 4th vaccine which is still also under investigation as the live attenuated version. We chose OMV because it has the outer-membrane proteins against which the antibodies are produced. And apart from that, there are no unwanted proteins which might cause unwanted reactions. So it's a specific set of proteins from the perisplasmic space, not the cytosolic proteins. In that terms, it has low reactogenicity, if you say.
But it's not a marketed vaccine yet?
Not yet. Investigational vaccine, I mentioned. So that was the choice, that it's a newer version and that we might be able to have a better immune response if we especially take it through the mucosal route. And that is the reason for considering OMV vaccine. Okay,
so then you go and spray dry this vaccine and you investigate stability. And in figure 3, I was quite surprised to see that at a temperature of 40 degrees, the non-stabilized vaccine is very stable. So is it in the end really worthwhile? Do you really need to do this spray drying?
Indeed, that's a very interesting observation, which we also had, that at 40 degrees, the liquid vaccine was not getting destabilized. However, this observation is only till a month. If you see the fourth line, which is a 60-degree liquid sample, on the day 1, it decreases by 80%, the Fach-8 antigenicity. So you cannot say by surety that at 40 degrees, okay, it's stable for 30 days, which might not be the case with further storage. Although if we talk about stability in general, it might look good for 40 degrees, it's stable. But if you consider the other parts, maybe the formulation, the processing, that might have some effects, but that was not investigated. But indeed, in just considering this graph, I would say we would require data for a bit longer study at 40 degrees to know the real nature of this destabilization.
Mm-hmm. Yeah, I'm always a little bit in doubt whether you can compare accelerated stability at 60 degrees with real life, let's say, because then, of course, you get denaturation of proteins. But anyway, but then I was kind of surprised in your earlier chapters. You investigated a lot of different conditions for spray drying. So you changed the stabilizer, the inlet temperature, buffer, all kinds of conditions. For this vaccine, you used only one condition. Why was that? And how would you go about if you had more time?
Yeah, so for this vaccine, when we initially spray dried with our tested drying conditions, we got a product that was quite stable. In that perspective, there was no reason to test more drying conditions because we had an RMC below 3%. That is good enough for storage. And the product was stable, at least for the antigenicity we checked, and also was immunogenic after the animal study. However, if I had more time in respect to changing things, we used trehalose as an excipient for making this formulation. What we observed was this formulation from trehalose is quite hygroscopic, and if you really want to deliver the powder, there are chances that it will aggregate. But again, the device that we were studying using, which we have also worked in the past, has also been shown in literature that the delivery of the powder is more in the trachea region and not really goes in the deeper lung. Mm-hmm. So that would require consideration.
I would like to go briefly back to the stability point, because you measure antibody responses against different components which are present in the vaccine. You see a little bit differences between the different proteins, but which ones would you consider as particularly important for the protection against pertussis in real life?
Of course, there are some, to say, star proteins which we would like response against. If we see the MS data, which is in the supplementary figures, the Fach-8 protein is the most abundant one.
The what, sorry?
Fach-8, so BAG8. That's the most abundant protein in this vaccine. Mm-hmm. Close to
is
50.
It also most important for protection against pertussis?
And at the moment, the literature has indicated that it's being involved, but if it's the major or the most important one, then I'm not completely sure. Mm-hmm. We also have, if you see the responses against BRN, which is another very important protein, and PTX, the pertussis toxin, and FHA, and we did mention all these, we did check the response against these proteins. So yeah, the most important proteins we tested against, I think. Mm-hmm.
Brief look, I think I will give the word back to the rector.
Okay, you understood my signal, yeah. Mm-hmm. Then I call upon
Thank you very much.
Dr. Metz, representing the company Intravec in Bilthoven. Very welcome to you.
Thank you very much. Esteemed candidate, Mr. Gaurav, I would congratulate you realizing your thesis. I can imagine that it was not an easy task. It requires a lot of time, endurance, and also a lot of cooperation with other people. Maybe also a little bit of luck. But finally, everything is crystallized and fall into place, like you see here on the cover. But it's not done yet. I have a few questions for you. First, I would like to start with the technique spray drying. It might be an easy technique for preparing or producing tons of milk powder or for using the encapsulation of food ingredients. But how would you manage it to prepare final vaccines performed on a GMP, which is in very small quantities, and how can you make a final product of that using spray drying?
Certainly, Rovenant. Thank you for the question and your nice comments about the book cover, especially. I understand spray drying is a very well-known field for food products and is still quite investigated in case of vaccines. I would like to mention here, in terms of products for GMP purposes, a recent clinical trial with a modified version of spray-dried vaccine for live attenuated measles was tested in phase 1 clinical studies and was shown to be immunogenic. In terms of having a product with the GMP rules and regulations, there was a product in the market that was spray-dried insulin, Exubera from Pfizer. It was withdrawn in 2011 for not spray-drying reasons, but they do have the facilities to produce doses forming 1 to 3 milligrams of fill and finish. So the technology is still available and has to be optimized with, or to be collaborated with the vaccine versions. On the top of that, if you really want to have sterilization of the product, you can also use terminal sterilization with radiation or something. So it is feasible, it is used in the clinical studies, and I think it's promising in that perspective.
And how to handle with it, because it has to be in an aseptic production process, how can you handle with that?
So at the moment that what we were testing was still in a ML2 facility and it was not aseptic, there are studies available, not in terms of literature, but a lot of companies are investing in making the spray drying process in an aseptic area. It already exists for lyophilization, and if there is a clinical batch that was used in phase 1 study with a spray drying technique, I'm sure this can also be extended to other vaccines.
Thank you. Now I would like to focus more on chapter 4, which is the effect of formulation on spray-dried Sabin inactivated polio vaccine. So you spray-dried the IPV vaccine. What you see on page 84, sorry. It was the dehydration step, which has a huge impact on the D-antigen recoverage. The rapid drying process was performed at a fixed temperature of 110 degrees Celsius, at the inlet at least. Would you consider an adjustment of the inlet temperature to get a higher D-antigen recovery? Do you know the denaturation temperatures of polio virus?
Indeed, temperature plays a very important role when we consider polio. Polio vaccine, what we have is in a special conformation called as D-antigen, which is when administered, the neutralizing antibodies are generated against it. At the temperature of 56 degrees Celsius, there's a change in conformation from D to C, which we don't have the polio neutralizing antibodies against. So we were trying to have a drying condition in which the outlet temperature stays at least below 55 or around that, because although we don't see yet the changes, we wouldn't like to go in a direction where our outlet temperature goes above 55 degrees Celsius and changes this conformation from D to C. So,
thank you. There was also a considerable difference in the stability of the polioviruses between the serotypes. That can be seen in figure 3, 4 and 5a on chapter 8 and 9 and onwards. How can you explain why the poliovirus type 3 is less stable than the other serotypes?
Indeed. So every serotype behaves differently during drying, and that we already saw during the experiments. It is quite well known in literature that the serotype 3 of the poliovirus is the most, from the Sabin strain, is the unstable one. And despite efforts from different groups, we are still not able to get a completely stable dried product. There have been, of course, studies with stabilization in the liquid form with the addition of excipients. But as it's known that it's an intrinsic thing about the serotype 3 to be unstable, it's just like that.
And there's a lot of homology between the serotypes. Based on that, it cannot be explained. Do you have other reasons why that serotype 3 of Sabin might be less stable?
Yeah, that's a good one, because I'm not sure out of my head that I could give you one pinpoint reason of the exact reason of instability of type 3. Like, for example, when you have them all separately and you put it at a higher temperature, they will all degrade. But when we were putting excipients into it, some serotypes were better in their stability profile, so we were able to stabilize them, and that unfortunately didn't happen for type 3, and the max that we could stabilize was 50%. So indeed, I'm not sure why intrinsically it's unstable, but with the addition or formulation techniques, we could stabilize the other two, but not the third.
Then a final question.
1 minute.
Okay, 1 minute. You used the inactivated polio vaccine. How would you do it when you are using the oral polio vaccine?
You mean in terms of drying?
In terms of drying, indeed. How would you formulate it? Should it be at least? Should it be? How should it taste at least? Should it be sour, bitter, or sweet? Because you are using different components for stabilizing the sprayed products.
I will answer it from the back to front.
Yeah, can you give it a short answer?
So it should be sweet. And previously people used to give it in the sugar cubes for oral polio vaccine. And the part of handling is considered. Of course, additional considerations are required, but the most important is it should replicate again after drying. So your analytics has to be changed in terms of that. And apart from the formulation part is considered, that has to be tested. It's difficult to just predict it without saying, it's no magic.
Thank you very much for your opposition. Then I call upon Professor Olinga, Department of Translational Biopharmaceutics in the University of Groningen.
Thank you very much. Esteemed candidate, first I want to congratulate you with your very nice thesis, and also like to include your supervisors for this very nice work. As you know, I'm not an expert in spray drying. I'm more a biological guy. And in chapter 6, I found something which I knew the name of. It was infliximab, so I thought, let's discuss chapter 6 with you. And when I looked at chapter 6, I saw that you used a liquid formulation of infliximab as sort of control in a lot of experiments. And I also saw that in there was sucrose. Why is sucrose present in that formulation?
Hopefully, opponent. Thank you for your question. In terms of the control in chapter 6, which is present with the sucrose, the answer to that is it's a commercial formulation. So the commercial formulation of infliximab, which is available in a freeze-dried product, has 3 components: sucrose and the proloronic and the buffers. So indeed, if you want to make test it further, so we had 2 aims. One was to test another freeze-drying equipment that was, or the way, it is the LyoGuard trays, and for that we needed a positive control, and that was the marketed formulations, and that indeed had sucrose in it.
But wouldn't it be better to only use the infliximab itself without the sucrose, or?
If you really want to have a product better than the market, you should have a control what is in the market to have a better product, you have to compete the competitor. So that's the reason we have.
Okay. And so you use that formulation also to look at accelerated stability experiments. experiments where you looked at 60 degrees. I wonder, why did you take 60 degrees in 1 month for stability? Is that normal for using that with antibodies?
That's indeed a good question. Generally, I would have started with 40 degrees Celsius. However, the melting temperature for infliximab is around 65, a bit above. I don't know exact figure by heart. So if you really want to test the robustness of the product, we chose to go at the higher range. So 60 was still very close to that, and the product was still able to show the TNF binding ability, and that was the reason for the temperature range.
And when you saw after 3 days after the stabilization test, you saw that there was aggregation present in the infliximab liquid formulation. Do you know what it was? Is it infliximab itself, or is there any reaction with the sucrose, for instance?
I'm not sure exact nature of what was responsible for this, but there were visible white aggregates in the liquid, and we assume that is infliximab aggregating, not with the excipient, but generally.
Because
I don't know the exact nature of that observation.
I did some research on, Google is always nice, and I found that sucrose itself, because at high temperature it can go into glucose and fructose, and then maybe glycation can occur. Is it something you heard before? Is that a possibility of infliximab, that it can react with glucose or fructose and then maybe aggregate?
That could be the reason. If it has a reducing, the sugar have the reducing end, they could, of course, react with the amino acid and sugar reaction, Maillard reaction it's called, but I have generally been investigated mostly in the dried form, because drying, you see in normal food products as well, like, bread has and we did observe a slight change for the product, which was dried, but I didn't see any change in coloration. So I don't know the exact nature, but that could be a fact that there is some reaction with the sucrose.
So could it not be that maybe a better control would be than without sucrose then as a formulation with liquid? As for stability purposes?
Yeah, if you think that way, that could be a consideration to exclude sucrose from it, to know the exact nature, but we didn't, so that's the best we had at that moment.
Did you ever try, because the aggregation product that you had after 3 days, did you also do that in a TNF-alpha binding assay? Did you also test it there? It was still binding?
If I remember correctly from the top of my head, the binding ability was lost with the aggregated liquid control.
Because I wondered if that's, because you say that it's a biological activity test, so to say. Do you really think that's a biological activity test, or is it just testing the TNF-alpha binding?
To a precise TNF-alpha binding ability.
Yeah, so not a biological activity in a way. So
yeah, it's an indirect reference to it, but yeah, you're right.
And then coming back to the original, if I still have time for...
Yes.
Yes, okay, good. Going back to your original plan that you did spray drying, although you used both inulin and dextran. Why did you use these both? Why did you do these sugars? Use these sugars?
Well, the reason to use inulin and dextran was just based on that they have a really high glass transition temperature. So if you really want to have dried products, you would like to consider something which is much higher than sucrose, the TG. And inulin has around 155, and dextran in fully amorphous is even higher than 200. And that was the basic reason for considering these pharmaceutical excipients.
And what kind of result did you expect in the end? Why? I mean, did you think that in the end that inulin or dextran would be better, or why?
In the end, so the idea was that if you have a formulation with such sugars, and if you want to make further a tablet out of it, because somehow in the back of the head was having an oral formulation, and that could bear the process of granulation and tableting. That was not investigated yet, but another publication from Morer in 2016 showed that with freeze drying with inulin, you could further formulate into a tablet. And if you see in that perspective, inulin is a good candidate to bear the further processing.
And that would also be your ideal formulation in the end, when you look at spray drying, that you would do that with inulin? And would it be spray drying, or would it be freeze drying in the end, what you see as the most promising formulation? Yeah,
as we saw in the stability study, we did observe some modification with inulin So if I would have to spray dry the product, because the freeze-dried product was much better in the stability study. For stabilization with spray drying, it would take trehalose as a excipient. So, yeah.
So that would be optimal. Okay, thank you very much. Give the word back to the director.
Thank you for your position. Then I call upon Professor Huisma, the secretary of this committee, Department of Pharmaceutical Gene Modulation, the University of Groningen.
Thank you. Esteemed candidate, first I also want to congratulate you and your supervisors with the research described in your thesis. I think it's a great example of pharmaceutical research which combines both applied and basic research. In your thesis, you deal with formulations of proteins and viruses, and the aim is, as I understand it correctly, to store these at room temperature for a long time. My research is focused on gene therapy, and we use viruses and genetic material, and for us also storing at room temperature is a major aim, because most of the time we need to freeze our samples at -80, and then even then we lose activity. Now, related to this, and I'm switching gear a little bit, you're probably aware that there are also genetic vaccines. And these contain, as the name already mentions, genetic material. It may be DNA or a virus. In the case of DNA, we don't need a cold chain. We can isolate DNA from bones that have been buried for 10,000s of years, and still the DNA is there. So how do you see the future of vaccines? Do you think these DNA vaccines will take over? At this time, we already have 4 DNA vaccine products that are approved, not yet for humans, but for veterinary use. So do you think after 10 years we will only use those DNA vaccines, or is there still a future for the, let's say, the old-fashioned viruses and bacteria?
Okay, Robin, thank you for the interesting question. It's very speculative nature. So, coming back to the DNA vaccine and the traditional vaccines, maybe 40 years ago we talked about that we will have, considering oral polio vaccine, we had IPV, the inactivated polio vaccine, oral polio vaccine. We saw the side effects like, the paralysis caused by the vaccine, and we thought, okay, we will change it to IPV in the next 10 years and next 20 years, and that will be fine. But still, polio is not yet eradicated, and we still, although substituting one by one IPV into the chain to the oral polio vaccination. So in that perspective, I would say that there is still future of the current vaccination, which are the viruses and bacteria. Coming back to the DNA vaccine, which you said, indeed, the potential looks nice. But if you talk, I'll tell it in 2 ways. First is the stability part that you said that, okay, they are stable at room temperature. To comment on that, I would need more information. That is it just the DNA you're talking about? Is it the other formulation parameters which are inside, which anything can happen with a temperature fluctuation? So is it just the DNA which is stable, or is it the whole formulation that is stable at room temperature? Commenting on that, there is a slight, how do you say, from the WHO, a slight exception available. If you can prove your vaccine is stable at 40 degrees Celsius for 4 days, you can exclude it from transport outside the cold chain. And so if that is the goal, please go in that direction.
Okay, well, I'd like to move on. You're absolutely right. Is it just DNA, or do we need something extra? And in many cases, we indeed need a viral vector in order to get good gene expression, so protein expression, in order to induce a good immune response. In this regard, I think it's interesting that quite recently there is a vaccine developed for Ebola. And you're probably aware of this. This is the vesicular stomatitis virus that has been genetically modified to express the Ebola proteins. Now, if you would think about using your technology for genetic transfer, in your vaccines, you're looking at protein stability, basically. What we want in viral stability is having those viruses still able to infect cells and express proteins. So would that be an additional challenge in your technique?
So I think you're talking about the EBO-VAP vaccine from Janssen, which is, I think, in phase 2 or 3. But doesn't matter. The question is if I need modification in the vaccination, the vaccine itself. Of course, that's one of the challenges, that it should still replicate and infect. You could consider drying it if you really want to use a different mode of administration, inhalation or something. However, you can always simplify it. It's not drying, but you can always increase the stability by adding extra excipients and checking if those excipients just in the liquid state provides you that stability. That would be a bit simpler if different route of administration is not your goal.
Well, usually what we do with the route of administration is like the other vaccines, that we do an intramuscular injection. And then I think it would be nice to have a formulation that it is compatible. And then being stable at room temperature would already be an enormous advantage compared to what we have to use now. So coming back to your title, Drying Made Easy. How easy would it be if I would give you let's say an adenovirus, and I ask you to develop a spray drying technique?
Indeed, the title says Drying Made Easy, and in discussion... Can I finish the answer?
Yes, finish.
So indeed, I say it's drying made easy, but as I mentioned in the discussion, that for every vaccine candidate, you need a different process condition and different formulation. So although it can give you a direction, this thesis, the chapters that maybe you can start with here, but to get a final product, you really need to test them.
Thank you. The College of Deans will withdraw for some time to consider its decision.
je plaats.
Esteemed PhD candidate, the College of Deans of this university, represented by us after reading your PhD thesis and the accompanying propositions and hearing you to defend it, has decided to award you the PhD degree at the University of Groningen, which you desire. Your primary supervisor, Professor Freiling, is authorized to award you this honor in the customary fashion.
I'm happy to accept the task assigned to me by Director Magnificus. However, before performing this task, I would like to address a few personal words to you. Dear Gaurav, surely you understand that the words that I'm now about to speak are also on behalf of your other supervisors, Professor Gideon Kersten and Dr. Jean-Pierre Amaury. I guess it is somewhat like 7 years ago that we first met. You came as a master student to our department for one of your research projects. Coming from the University of Delhi, you had entered the prestigious top master of medical and pharmaceutical drug innovation here in Groningen. At that time, there were quite a lot of students in our department. But in one or the other way, you were always prominently present among your peers. You were able to distinguish yourself not only by doing good research, but also as an extremely relaxed but still very cheerful person. Moreover, Gaurav, you showed to be one of the bravest persons I ever knew. Surely it requires guts to go for a water ski trip when you cannot even swim. But still you did it. Together with your friend Douwe, you stepped on the board and halfway you capsized. You barely drowned, but together and with some help from Douwe, you managed to survive. Thank God. Already at that moment, I realized that you probably were one of those persons that has some extra talents in collaboration, or one of those smart guys that could make other people run for them. I wasn't sure what it was at that time, but there was time enough to find out. You finished your master studies quite successfully, and you even won a scientific prize at the International Student Congress of Medical Sciences at the ISCOMS. After finishing your master period, your student life was over and you entered a new phase in life. You had decided to become a PhD student and you were looking for a position. Your first proposal was rejected by the graduate school, but luckily, at that moment, Gideon Kersten and Jean-Pierre Amaury stepped in. For a long time, we knew them already as respected colleagues and good collaborators, both employed at what was at that time called the Netherlands Vaccine Institute. They had a PhD position available for a collaboration between the Netherlands Vaccine Institute, the University of Leiden, where shortly before Gideon had become professor, and our department at the University of Groningen. Until today, I'm very grateful to both Gideon and Jean-Pierre for their efforts to make this position possible. But I'm sure, Gaurav, I'm not the only one who's grateful to Gideon and Jean-Pierre, because you should be too. They chose you to fill this position. And so you moved from Groningen to Bilthoven. But probably that move was the smallest one you had to take. Because when after being a master's student, you become a PhD student, there are huge changes, and a lot of changes. As a PhD student, you have to organize and carry out your own research. You have to design your own experiments, and you have to write your own papers. But you picked it up, and you made a swift start. Also because Jean-Pierre had organized a kind of a commercial startup project. You started with it and it became the last chapter in your thesis. So how did you write your thesis? Backwards? Remarkable. But you started with infliximab. And you set out to become a good scientist. Your research has always been helped by the nice research meetings that we had with both Gideon and Jean-Pierre, and many stroopwafels. Gaurav, when I look back into your TSP, it's clear what research is. Actually, research is nothing more than you plan a lot of things in detail, ended up with doing completely different things instead. That was also with your research the case. And soon after you started, you were doing exciting things in drying new vaccines, things we had never thought about before. But that's good research. Not many people probably remember what happened then. But those who probably think would understand what happened. Immediately after the Netherlands Vaccine Institute hired you, the Serum Institute of India took it over. They knew what smart guy from India had come there, so it was a good reason to come over. Surely these guys in India must have known. But bad luck for them. The research department was not part of the deal. So you remained a Dutch civil servant employed at the University of Groningen, working in Bilthoven. Surely becoming a doctor is not an easy journey. And even an extremely relaxed person like you are may encounter problems and difficulties. To begin with, good research is 80% failure, and those of us working in the lab know that the 80% is an underestimation. Writing papers is difficult, and sometimes you need help. And surely you did have your share of personal misery too. I think we all regret that, for example, today your father can't be present here. However, Gaurav, you managed, with the excellent guidance of Gideon and Jean-Pierre, and some help from Leiden and Groningen, you ended up with a great thesis, Drying Made Easy. 5 publications can already be found in PubMed, and some others to come. A great achievement, and something you can be proud of. You have developed to become an adult researcher, and from today on, you may call yourself Dr. Kanoya. But who's Dr. Kanoya? I still had that question from the start. I already mentioned it at the beginning. Surely you are what in English would be called a smart cookie. A big stroopwafel, I guess. Here it is. That's what you are. In Dutch we would say probably een handige jongen, because that's what you are. And indeed, you are able to make people run for you. You're a great Bollywood dancer. I just saw the movies from the Congress in Granada. They're all over my department. Gaurav, Bollywood dancing, great. You're a guide in the city of Utrecht. But above all, Gaurav, you remain the cheerful person with excellent communication skills that has all reasons to be proud of himself and your achievements, and with, I'm convinced of that, a bright future ahead. Dear Gaurav, I would like to thank you for the excellent collaboration that we had over the past years. Your contribution to our scientific progress and the way you were moving between Bilthoven, Leiden, Groningen. You really enabled the collaboration, the good collaboration we had, and I thank you for that. I wish you all luck and happiness in your future career and personal life. I also want to congratulate your family, especially your mother, who came from India to be present here today. It's great. And in a few moments, and I wish you for today a great day, because, as you rightfully quoted the Dalai Lama in one of your propositions, happiness is not something ready-made. It comes from your own actions. So make it today. Ladies and gentlemen, I would like to go to the official promotion, so may I invite you to stand up. By virtue of the powers vested on us by Dutch law, in accordance with the decision of the College of Deans of this university, I, Hendrik Willem Frijlink, confer on you, Gaurav Kanoya, the title of doctor and all the rights associated by Dutch law or custom with the PhD degree at the University of Groningen. Do you, as a doctor, promise to act in accordance with the Netherlands Code of Conduct for Scientific Practice? I do. In that case, it's my great pleasure to be the first to congratulate you on this diploma that you get. You can all be seated again. Thank you, Mr. Rector.
As proof of this PhD degree, I will present you with this certificate signed by the Rector Magnificus, Secretary, the two supervisors and the co-supervisor, and sealed with the Great Seal of the University. But before doing so, very learned sir, it is my pleasant duty to congratulate you also on behalf of the College of Deans of this university on the degree you have been awarded. And here we have the certificate. We also have the certificate of credit school to present you, and the album Amicoid. Thank you very much. And now I declare this ceremony for closed.
Yeah, it's true, but I don't have much experience on that.